This video is on question 31 from the 2013 IMAT paper. So in this question we have to complete the sentence of the direct product of transcription of recombinant DNA can either be insulin, monoclonal antibodies, mRNA, the primary structure for a protein, or a replicate DNA molecule. So first things first, let's make sure we actually know what transcription is. So what is transcription? Well, transcription is the process of copying a section of DNA, in this section being called a gene, into mRNA. And so below I've drawn a diagram of this. Firstly, we have our DNA here, with the DNA bases drawn in blue and the green dashes between them being hydrogen bonds. Then, DNA helicase breaks these hydrogen bonds to allow the DNA strand to separate, and then a new RNA strand composed of bases which are complementary to those in the DNA strand gets built up, with the RNA polymerase catalyzing the joining of each subsequent nucleotide. And so when this is complete, this RNA strand is called mRNA. That's messenger RNA, otherwise known as a transcript. This mRNA transcript can then be translated by a ribosome to produce a polypeptide. The polypeptides can then be processed and assembled together to produce proteins. So now, upon looking at our answers, we can see that it's not insulin, as insulin is a protein and is therefore a product of translation, not transcription. Next, monoclonal antibodies. Antibodies are also proteins and so therefore a product of translation, not transcription. And then we have mRNA, which is our correct answer, as we know that transcription is the copying of a section of DNA into mRNA. And for completion, it's not the primary structure of a protein, as that is what is produced directly out of translation, as the primary structure is the sequence of amino acids. And finally, a replicate DNA molecule. Now we know that's the product of DNA replication rather than transcription. So this corresponds to answer C. And just as a side note, all recombinant DNA is the product of experimentally modifying an organism's DNA so that it incorporates another organism's DNA into its DNA. And so the product of transcription of recombinant DNA will still be mRNA. So this recombinant DNA part is just to try and throw you off.